ought to be confirmed by observation. And from this very minute, we will start our journey together to seek this truth through various aspects of research in the field of botany. Welcome, everyone. Thank you for joining us for today's webinar. My name is Dr. Sandipan Ray, and I am the Assistant Professor at Department of Botany, Durgapur Government College, under Kadji Nazrul University. Along with me is Dr. Anish Bhattacharya, Assistant Professor at Department of Botany, Durgapur Government College, and we will be your host for this 2 days webinar. Today, we all gathered here to witness how plant science are evolving in the crucial moment on 25th and 26th April 2025 through DBT Star College Scheme sponsored 2-day national webinar on plant diversity to genomics, emerging trends and future direction, organized by Department of Botany in collaboration with Internal Quality Assurance Cell of Durgapur Government College. We have participants from all over India, like from Andhra Pradesh, Gujarat, Rajasthan, Orissa, Assam, West Bengal. Let me go through the schedule for today. This webinar has been divided into three main sections. And to start with, we'll have an inaugural session, followed by plenary session, where Dr. Sandeep More will deliver his thought. After that, we'll have a lunch break, followed by paper presentation. Our intent is to spend most of the time to nurture the nature through this 2 days scientific gathering in virtual mode, where leading academicians, scientists, researchers, and most importantly, students will exchange share their experience and research on various avenues of botany, covering diverse group of plant kingdom. Now, without further ado, we will turn the time over and we'll start the inaugural session. Um, our respected principal sir, as well as he's the honorable chief patron for this webinar, Dr. Devnath Balit, Durgapur Government College. Uh, he is busy in some academic work, and so that he's not joining us today, but he convey his best wishes for the webinar to be a successful one. So thank you, sir, for your message that you conveyed to me. Now, I want to request our Honorable Vice Patron, Dr. Nivedita Acharya, Coordinator IQAC, and also the Coordinator of DBT Star College Scheme, Durgapur Government College, to kindly address the gathering. Madam.
Yeah, Dr. Shandipan, am I audible to you?
Yeah. You were audible, madam.
Yeah, okay. First of all, thank you for giving me the opportunity and inviting me to this August event. I would like to thank, first of all, Department of Botany under the able leadership of Dr. Ashok Bhattacharya for organizing this wonderful event, which actually portrays the admixture of scientific understanding as well as giving a platform to the students for showcasing their scientific understanding as well as different participants through paper as well as poster presentations. I am very much thankful to the departmental coordinator of DBT Star College Scheme, Dr. Shundipan Ray, for always extending his help and support in organizing such, say, enriching events for the students. The Department of Botany has been organizing a diverse scheme of events. This may be new experiments, this may be workshops, this may be their invited lectures to ensure expert interactions with the students, which is one of the important facets of DBT Star College Scheme of Department of Biotechnology. We owe everything to this Department of Biotechnology for giving us the opportunity and the funding for organizing such events for improving the analytical, the practical, the subject understanding skills of our students. I am also thankful to all the faculty members of Department of Botany for extending their support and cooperation. My sincere thanks goes to the resource persons who will be delivering their sessions in this 2-day seminar. or better, I can say the webinar to be more precise for the online. And for the students, I would like to say we are organizing these for the sake of you so that we can cultivate the creative impulse among you. Because science deals with diversity, which goes along with subject understanding as well as practical understanding, problem solving, and critical thinking skills. So it is a great opportunity for you students interact with the resource persons as far as for making your topics clear. So many thanks to the Department of Botany for organizing them or for organizing such a wonderful event, which will be very helpful for improving the skills of the students. So with this, I would now like to request Dr. Shondipon Ray to go with the technical session. Dr. Shondipon, please.
Thank you. Thank you so much, ma'am. Your presence and wise word will help us to magnify the trust, actually, for the better world in a possible way. Now, our next speaker is Dr. Shubhajit Oja. He is the TCS of Durgapur Government College, but due to some administrative reason, he's not with us at this moment. So, again, he conveyed his good words and he wishes success for the program. He's joining us in few minutes actually. So without further ado, I will humbly request Dr. Ashok Bhattacharya, Head Department of Botany, to address the gathering. So
Okay, am I audible, Sandeepan?
yes, sir, you're audible,
Yes.
sir.
Okay. Thank you, Dr. Sandeepan Ray sir, for giving the chance here. And now, actually, first of all, I appreciate the presence of all the students and the distinguished gatherings. Good morning, everyone, and welcome to all the participants here in the national level webinar on the particular subject entitled, you know, that the plant diversity to genomics, emerging trends and the future direction. And this national level webinar actually is organized by the Department of Botany in collaboration with the IQSC, Durgapur Government College. Our respected coordinator, Madam Dr. Nivedita Acharji, is present over here. And thank you, Madam, for your presence with us and to illuminate something in favor of and towards the distinguished participants of this seminar. And thank you, ma'am. And your actually comments are also very immenseful. And particularly this national level webinar, it consists very good title, that is the from plant diversity to genomics. That means from classical aspects to the plant molecular biological aspects, and what are the different trends of the research and the trends of the academics are going on, that we will know in today's different lectures, the plenary lectures provided by different distinguished scientists throughout the country, and who are reputed themselves not only in India but in abroad also. We have got some distinguished scientists, and definitely we are very much thankful to them. So before that, actually, I welcome to all the participants, all the dignitaries, and all the guests in this national level seminar. And this national level seminar in form of the online, that's why it is webinar. It highlights the importance of the particular different themes. Those are incorporated in the plant diversity and the genomics study, that incorporated the different levels of the different organizations of the subject, the botany and the plant science as a whole, starting from the classical, the systematics part, towards the molecular biology, even the plant physiology, ethnobotany, in every aspects we have the different dimensions, and we have obtained many research papers, significant research papers in this particular endeavor. Those are very much highlighted, and those will be reflected in front of all you the participants. So the thank you all, particularly to the participants, my beloved students of this department who are present here right now, and special thanks to the organizers of, particularly the organizers of our department, Dr. Sandipon Ray, Dr. Anish Bhattacharya, Dr. Prashanto Saha, Dr. Subhajit Ojha, all faculties, Dr. Even myself as a total, the head of the department, myself, Dr. Ashok Bhattacharya, and by boosting up of all the youngsters, this I have also I am very delighted by boosting up all the youngsters. Those I mentioned the name of the Dr. Ray, Dr. Saha, Dr. Bhattacharya, Anish Bhattacharya, and Dr. Ojha. All of them supported, and I'm very much thankful to all these organizers. And not only to these organizers, I'm also thankful to the coordinator ma'am, DBT coordinator ma'am, because the DBT coordinator and the IQAC coordinator, Dr. Nivedita Acharjee, associate professor of the chemistry department, actually she has also extended her hands to organize this type of the national level webinar. So as such, we can exchange the different thoughts and ideas throughout the India, because we have got many papers from outside of the West Bengal. That's why thank you, madam, for giving your cooperation towards us and sponsoring. And that's why this webinar is actually sponsored by the DBT Star College scheme. So I'm very much thankful on behalf of the Botany department to the sponsors and other supporters. Not only this, we are also very much thankful to our head of the institute, Dr. Devnath Palit. Always he has boosted up and he has encouraged us to organize such type of the events always, and that's why we are very much thankful to have such an highly academics and a highly dynamic administrator come academicians, Dr. Palit, who always help us and always cooperate us. So we participants are productive and the engaging experience, the best wishes. I convey my best wishes to all the participants for a particular productive and the engaging experience. So I am honored to welcome you all to participants, dignitaries, chief delegates, guests, everyone who have contributed their findings in our national webinar, and that's why I welcome to you all to the Durgapur Government College for participating in this particular theme of the seminar, that is from plant diversity to genomics. So I appreciate the efforts of the organizers, and I thanks to all the sponsors and the supporters. I wish all the participants a fruitful experience for engaging discussions, meaningful connections. So let's make the very good opportunity to discuss everything. And this today's national level webinar, the plant diversity to the genomics, I already mentioned that the particular topic of this subject, that is the plant diversity to genomics and the emerging trends and the future directions, that consist of the various sub themes, starting from the classical to the molecular biological field, the plant genomics study, everything. And we got a number of the more than 12 research abstracts in the various fields, in the particularly the ethnobotanical studies, ethnobotanical of the medicinal plants, review report of the dynamics and the implications of the climate-driven disruptions in the plant microbe interactions. So the roots of the healing, that means the traditional plant wisdom to the knowledge. So not only the ethnobotanical aspect, we got the different papers in the plant physiological aspects also. That means the unveiling of the new species of the Amantia through the morphomolecular taxonomy, molecular taxonomy, ethnobotany, plant physiology, allelochemicals, that means the weed warriors of the nature, allelochemicals, bioherbicides, adaptive responses, so the green solutions of the plant-based water purification, many such type of the papers we have got. And not only this, the plant physiological aspects, the particularly the chromium toxicity, the sulfur toxicity, and the cadmium toxicity, all these papers we have got. And the different papers related to the pharmacognosy related research, that is the alkaloid potency, cancer biology, and some evolutionary aspects, the floral divergence and the evolutionary aspects in the sky islands, ethnobotanical aspects. So many papers, more than 12 research papers we have obtained, and you will be happy to know that we have the most distinguished professors of the various institutes from Presidency University, Guwahati University, Kollani University, Murshidabad University, and they have given their consent. They have given their kind consent to share their vast experience, to share their knowledge on the particular subject of the botany, and the vast experience to share with our students, so that our students can be benefited, and they can get some ideas about the new researches what are going on, because the students nowadays they are doing the that means the honors 4 years honors program following the new education policy system, and after completion of the 4 years honors, they will directly, after qualifying the NET or the GATE examination, they will be directly enrolled as the PhD students. So at the time of the undergraduate studies nowadays, it is very much essential to know the different research fields, and to sensitize the research areas, in what different types of the research areas are going on, because the students, you know, that in your 7th and the 8th semester program during your honor study, you particularly develop certain research projects and the dissertation projects, and the dissertation projects, entirely projects, it will be developed, dissertation paper, it will be developed exclusively by following the research work. So for successful completion of that dissertation work from nowadays, you need to know what are the different research areas are going on, what are the different particular research directions are going on, and that's why we have decided to organize such type of the event, so that your knowledge could be enriched, and you will be sufficient enough in which particular direction you will follow your work. So that is the main motto of our webinar. Our main motto of our webinar is to not only to gather the distinguished scientists in front of you, not only to to talk with each other, not only to gossip with each other. Actually, we want to motivate you, dear students of our department. We want to motivate you that how the researches are going on in the different aspects of the subjects, and you will be motivated by the teachers of your department. Simultaneously, we will be motivated in this seminar by the other distinguished professors and the other distinguished research scholars who have submitted their research papers to this webinar, and they will present the research papers during the 2 days national level webinar, particularly the post lunch session, 2 days post lunch sessions. The different research scholars, they will present their research papers, and similar research paper students, I am saying you that after 1 or 2 years you have to present such type of the research paper. So until and unless you get some idea from right now, you will not be able at that time. If you are not able, so after 4 years completion of the research UG courses, no institute will select you if you don't have any ideas about the researches what are going on right now. That's why, to motivate you and to encourage you, we have decided to to organize such type of the national level webinar so that you get the idea in which direction you will follow your work. Many of you, somebody of you, some may be interested in the molecular biology, someone of you may be interested in the taxonomic research, someone of you in cytological or the molecular biology, biotechnology, ecology, phytogeography. Many fields are there. So what particular field you will choose, that is the most important thing. And that's why this webinar is conducted. So you listen it very carefully, the students, all their students, that the what type of the different researches are going on, and the four distinguished professors of the different universities, like our respected Madam Professor Mousumi Poddar Sarkar, Professor of the Life Sciences, Presidency University, a distinguished professor in the field of the lipid chemistry and the graphite, particularly taking the plant materials, the graphite, lipid chemistry, biochemistry, distinguished biochemistry, internationally famous. She will deliver the lecture in presence of you tomorrow. Professor Neera Sen Sarkar, a distinguished professor, associate professor of the Department of the Botany, University of Pollani. Madam Neera ma'am, I know her personally because Neera ma'am has a wide knowledge in the subject of the algae. In the subject of the algae, Neera ma'am has directly performed many research activities directly in the Sundarban area. So under the leadership of the Professor Kumud Ranjan, late Professor Kumud Ranjan Noskar. So the Neera ma'am will is the first one in India who has developed the tissue culture technique, and particularly I know that the Neera ma'am is for the first time in India who has developed the tissue culture technique for cultivation of the diatom. Diatom সাধারণত টিস্যু কালচার করে করা হয় না। নীরা ম্যাম হচ্ছেন প্রথম যিনি কল্যাণী ইউনিভার্সিটিতে ডায়াটমকে টিস্যু কালচার করে প্রোপাগেশন করার জন্য ফার্স্ট she has developed the lab, and she has published the work. I know her personally. So the diatom টিস্যু কালচার করেও যে ডায়াটমকেও কাল্টিভেট করা যায়, তোমরা ডায়াটমের নাম শুনেছো, এটা কিন্তু নীরা ম্যাম করেন। সো these different types of the algal research, algal biotechnology, nowadays the algal technology research is very much important for modern days perspective. So then she will exchange her wide knowledge on this particular subject with you, and you will be benefited, I think, more. You will be more benefited. Not only this, you will be another different, that means the internationally and the nationally reputed scientist you will get, that is today after a few minutes you will listen the lecture provided by Dr. Sondip More. Dr. Sondip More, he's the professor of the assistant professor of the Murshidabad University, who has done a lot of work in the field of the palynology and the forensic palynology, the most applied aspects of the palynological and the other related works. And not only this, another very good researchers and the scientists you will listen the lecture tomorrow, that is the Professor Dr. Arun Kumar Datta, assistant professor of the Department of the Botany, Guwahati University. So he has the vast knowledge in the subject of the mycology and the plant pathology, and many new species are being day by day being developed by Arun sir, Dr. Arun Kumar Datta from the Guwahati University. So Arun sir will share his excellent knowledge and the experience with you, the how the mycological research can be carried out, and it can be taken for your further research and the further academic pursue. So all these lectures you will obtain, students, and you will be benefited a lot from many lectures, from many abstracts. And you will be happy to know that a few ex-students of our Department of the Botany, they are also participants here in the webinar. They will present their findings, research findings in front of you, so that you will be boosted up more and you will be encouraged. And I think that the most of you students Here the student participants that you will after 1 or 2 years you will also come and follow the same type of the platform, so that the new frontiers of the research can be carried out. And I am very much thankful to all the organizers, and particularly to all distinguished professors who have given their kind consent to be with us and to share their vast knowledge with our students. And I am not only like that, I am also thankful to all research scholars here. Our in-house research scholars are also here, and the research scholars who have presented paper here, I'm also thankful. And because they also are very much willing to share their knowledge with our students and the participants and all the distinguished botanists to develop some new insights into these particular subjects of the plant science. Thank you all. I'm very much thankful, and I personally thanks to all of you, and I personally welcome from the on behalf of the Department of the Botany to you all. Thank you very much.
Thank you so much, sir. And it means a lot that these words are coming from you, especially from our own beloved head of the department, Dr. Ashok Bhattacharya. We have summarized everything. There is nothing left for us to do, and he made our job quite easy for this 2 days webinar. Now, I will directly go into the technical session. I will like to request my colleague, Dr. Ashok Bhattacharya, Dr. Anish Bhattacharya, to please carry forward this webinar with the technical session and introduce our first speaker of the day, Dr. Sandeep.
Hello, am I audible? Dr. A?
Yes, you are audible. You are
Yeah.
audible.
So do we have our respective speaker here already, or he's not there? Okay. Okay. With the permission of Dr. Ashok Bhattacharya sir, our HOD, and
Huh?
along with, yes sir, can I introduce him?
Yes, 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 yes.
Okay,
Introduce.
okay, 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 okay. So it is a great pleasure for me that I am introducing Dr. Sundeep More here because Dr. More, I have been knowing him for many years, and he is like an elder brother for me, and he is always motivating me from the day one of my struggle period, and still now he is supporting, he is advising me for many aspects. However, let me introduce him to you who actually don't know him. Dr. Sandeep More is currently working as an assistant professor in the Department of Botany, Murshidabad University, Bahrampur, Murshidabad. He did his BSc in botany from Sripot Singh College in 2005. Then he completed his MSc in botany with specialization in paleobotany, palynology, and teleology from University of Kalyani in 2007. He was awarded a PhD degree.
Chemical composition in their cell wall. A chemical complex, a copolymer of beta carotene xanthophyll, especially anthraxanthin, and fatty acid, which comprises the outer wall of the pollen. And it makes it highly resistant to decay. Any extreme temperature, any extreme pressure, the pollen can survive, can tolerate. But except two chemicals, that is oxidants, that is hydrogen peroxide H2O2 or CrO3, these two can damage pollen grains. So if I give any pollen on a slide and set under microscope, and if I ask my student that can you tell me which taxa is it? Is it hibiscus pollen or coconut or rose or lychee or whatever? Which taxa, which pollen taxa is it? If my students does not know the characteristic feature of every pollen grain, they would not be able to identify the plant by the pollen grain. So to identify a pollen specifically and surely up to taxa, up to genus or species, we must have to know these 7 characters of a pollen grain. What are the characters? Pollen unit, pollen polarity, symmetry, shape, size, aperture, ornamentations. So there are 7 characteristics. As my time is not very less time I have, I am not going to the details of the pollen characters because this is not today's context. I would just tell you the story of the applications. So these are the characters that we have to study and we have to get expertise on it. And for those characteristics, each and every pollen taxa on earth is unique, as no two person, each and every people has their unique fingerprint. Each and every human being has their unique DNA. And my DNA is not, it cannot be matched with any other person. My fingerprint cannot be matched with any other person. Just like that, each and every plant taxa has its unique pollen character, and that helps us to identify the plant up to its genus or species. And that's why pollen grains are called nature's fingerprints. And this is the reason that pollen grains are used in various types of applications. Today, I will just tell you some glimpses of the different applications. So this is the scenario. If we divide palynology, it is mainly divided into two branches: geopalynology and neopalynology. Geo means pollen study from earth or soil or rock or fossil, and neopalynology means modern pollen study, that is living pollen. It may be from fresh flower, it may be from honey, it may be used in forensics to solve the criminal cases, it may be in to detect the allergic pollen grains which is present in the air, or other branches that I will go further. First application I will tell you, that is copropalynology. Copropalynology, if we want to know, first we have to know what is coprolite. Coprolite is the fossilized animal excretion, animal dung, animal excretion which got fossilized. We call it coprolite. And if we study pollen grains from the fossilized coprolite, we are able to tell precisely the diet of the ancient animals, which were herbivorous. So already so many different from India and from abroad, coprolites have been studied, and different animal diets have been established in all over India, all over world. Especially in India, if I mention, it is in BSIP, Birbal Sahni Institute of Paleobotany in Lucknow. They work on copropalynology, and if you go to Google, you will get multiple publications on copropalynology. And that help us to establish the diet of the ancient people, those who have got extinct today. Next is archaeopalynology. Archeo means we all can understand ancient people, ancient civilization. For example, it might be Harappan civilization, Mohenjo-daro, Indus Valley, Egyptian civilization, and so on. So any civilization which existed on Earth 2, 3, 4, or more thousands of years ago, if we get ancient pots, ancient potteries, or any part from those areas, those cities, ancient cities, and if we can be able to study the pollen grains from those pots, so we can tell you that 2000 or 4000 years ago, those people who existed at that time, what was their food habit? What type of crops they used to cultivate? Obviously, it was wild variety. What types of vegetables, fruits, cereals, pulses, what they used to cultivate, as well as what type of plant they used to cultivate for their um, animals, household animals. So this is also possible by the study of pollen grains from ancient pots. This story I always tell. Just one second. Am I visible?
Yes, you are. You are visible.
Okay, so I always tell this story to all my in all my lectures and to my students because they should know that pollen grains are not we should study because it is in the syllabus in our graduation or post graduation. It has various applications beyond our imagination. So this is one of the example, and it is called the mystery of Shroud of Turin, which was revealed by the study of pollen grains. What is Shroud of Turin? It is believed that after the death of the Holy Jesus Christ, his holy body was wrapped in a cloth and then it was graved. So the cloth is called Shroud of Turin. And that Shroud of Turin was kept in a museum which is called Cathedral of St. John the Baptist, Italy. Millions of people every year visit that museum only to see this holy cloth called Shroud of Turin. But few years ago, people questioned this cloth and they asked that what is the surety, what is the authenticity, that this is the actual cloth which was used to wrap the body of Jesus Christ. They can lie, it may be untrue. So the cloth was sent to famous European paleontologist Max Free, and Max Free washed the cloth, got the dust and pollen grains from the cloth, studied the pollen grains under microscope, identified them, and he got 49 different type of taxa, plant pollen taxa, which actually the plants, those which grow in the Mediterranean, Western Mediterranean countries like Jerusalem, Israel, or surrounding areas. Now tell me how the pollen grains of the plants from absolutely different phytogeographical region of Western Mediterranean country or Israel came to Italian museum's cloth. Some people may tell that it is possible that the cloth was brought from Israel, but the cloth may not be the actual cloth which was used to wrap the body of Jesus Christ. The question may come, but the thing is that it was at least proved that yes, this cloth came from Israel. Hence, they proved that it was the holy cloth of Shroud of Turin, which was used to wrap the body of Jesus Christ. And the mystery was solved after many, many years. Next story I also would like to share with all of you. It is called The Mystery of Chinese Terracotta Army. I am sure that most of you have seen these photographs in National Geographic Channel or Discovery Channel. In China, there is a province called Lingtong Shanxi, where thousands of terracotta army and terracotta horses were hidden underneath the soil, underground. And when some archaeologists were digging, when they were excavating, suddenly they found that terracotta army is hidden, and they kept digging, and they got numerous terracotta army, thousands of terracotta army, which was dug underneath the soil. So it was a big mystery in the world that what is the story behind it? Why these terracotta army and horses were made and dug in the underground the soil? So there are three samples were taken from the location: soil sample from the terracotta army, soil sample from the horses, and also soil sample from the location where it was dug. And it was sent to the paleontologist for the pollen study. Paleontologists found very interesting story. They saw that herbaceous pollen, that means lower plant pollen grains, were dominant in the warrior or army samples, whereas the arboreal pollens, that means tree pollens, were predominant in the horse samples and the locations where they were dug. So it was proved that as the warriors were more in number than the horses, warriors were produced hundreds of kilometers away from the location, and they were dug, they are brought here and dug inside. You may ask me a question, why they were dug? It is a very funny story. it is not believable to anyone. The last independent king of China, he thought after his death, when he will die, his kingdom will be taken over by the other kings. So he dug terracotta army and terracotta horses all around his kingdom. And he thought when he will die, these terracotta horses will get alive and they will protect his kingdom. So he was absolutely mad, and that's why he did this. But the mystery that where from it was brought, it was solved by the study of pollen grains. This image, if you see, you know that there are multiple pollen grains in the air, among which some are allergic, allergenic, which cause allergic reactions in our body. When we inhale the pollen grains by our nostrils, it may cause headache, runny nose, itchy skin, etc. etc. So, why these pollen grains cause allergic reactions to our body? The cause is we all know that pollen grains they have some proteinaceous substance, and our body when they enter our body produce some release some antibody immunoglobulin E. That antibody and proteinaceous substance they produce a chemical compound which is called histamine, leukotriene C, bradykinin, and etc. These are very allergic and they cause allergic reactions to our body, which is called pollinosis. And this type of allergic disease, these are named as rhinitis, dermatitis, etc. etc. So we all know like Parthenium, it is a weed around our locations, and in European countries and all over other places, some common allergic pollen plants are poplar, alder, timothy grass, ragweed, scotch broom, etc. Cell wall. A chemical. Next branch of palynology, it is not very commonly known. It is called latropallinology. That means use of spores and pollen grains that may be useful for human health, that may be used as a medicine. And
Ought
it
to be
is
confirmed
believed
by
that
observation.
Arabian
And from this very
physicians
minute, we will start our journey
use
together to
Lycopodium
seek
spore,
this truth
commercially called
through
vegetable
various
sulfur,
aspects of
for
research
the
in the
treatment of stomach disorder, and also spores of Adiantum philippinense are used to cure cough, and the pollens of Cycas sarcinalis they have also narcotic properties. So we can see that pollen grains can be used as medicine as well. And in the ancient classics, it was also mentioned that in Vasant Desika, the great Vaishnavan saint said about the refreshing pollen bath by Lord Shiva to Lord Vishnu from the Indian laburnum, which is called Cassia fistula. In Bengali, we call it Kotolati. The pollen was used to mix in the water for a refreshing bath. And Kalidasa, in his poem Megha Sandesa, he alludes to the use of pollen grain of Lodra flower. The scientific name is Symplochus spicata, white hazelwood, as a cosmetic powder. So it was in ancient classics also, in modern day also, it is proved that pollens have their medicinal properties. This photograph, if we see, we can clearly understand that it is honey, and the study from honey, pollens from honey, it is called melisopalinology or melitopalinology. So, if we study pollen from honey, what we can do? We can do many things, but especially two major points are there. First of all, a commercial honey in the market is 300 rupees liter or 400 rupees liter. But if you buy a medicinal honey that is called neem honey, tulsi honey, sorse honey, azadirachta, or kalmeg honey, it will cost triple or 4 times more. So, as a common people, they are not able to verify that it is actually medicinal honey or local honey. But as a student of botany, we can verify it if we take one drop of honey on a glass slide, observed under microscope. If we get the pollen grains of neem, azadirachta, we will be able to tell you that yes, it is the neem honey. But if we don't get the pollen grains of neem, Azadirachta or tulsi, then it is definitely a adulterated honey. So we can detect the adulteration of honey by the study of pollen grain, first of all. Secondly, we can study the honey, and if we identify the pollen by up to its taxa or plant, we can tell the apiarist that these plants are preferred by the honeybees to produce honey. So if you cultivate or if you uh, uh, make apiculture near this type of plants, then you will get more amount of honey, which is financially profitable for them. Here I would like to show you this publication. I published this paper in 2021 in journal Bee World, and this is from the I have reported first time in India. You will be surprised to know that this honey is called Tetragonula iridipenis. It is called APD or Melipony. Common name is Melipona. Do you know these honeybee do not produce any beehive? What do we know? We know that honeybee, they visit the flowers, they take pollen grains and nectar, they produce a beehive, and they keep their honey into the beehive. But these are the honeybee, they do not produce any hive. Instead, they make hole in underground. Under the soil, they make hole and they keep their honey under the soil, or they make hole in the tree and then they keep their honey in the hole of the tree. This type of observation never happened in India before our publication. We published this, and if I show you this video, you will get to know it. Here you see every moment the honeybees are coming, going inside the hole, keeping the honey, and again coming out. So these honeybees are called stingless honeybee because if they bite us, it will not pain. Next topic and last application of my talk, that is forensic palynology. What is forensic? What do we know about forensics? We all know that forensics means anything that is criminal or that is legal. That means is which solves a civil or legal problem. Trace evidence tells us where a suspect or victim or object has traveled from. It may be firearms, it may be handwriting fraud, it may be bite marks, fingerprint, shoe print, face detection, etc. etc. But for the first time in India, I did my PhD in forensic palynology, and I have introduced this subject in India where I tried to solve murder, rape, accident, this type of problems by the study of spores and pollen grains of plants. Is it possible? Is it believable? Maybe not. But here you see the concept of forensics is based on a only single principle, which is called the exchange principle. The exchange principle or the locus contact trace principle. What this principle say? This principle say that everywhere you go, you take something with you and you leave something behind. So when a crime happens, the criminal will leave something to the crime scene and take something from the crime scene. And this principle help us to do what? To do connect these four things. To connect the victim with the crime scene, to connect the crime scene with the evidence, to connect the evidence with the suspect, or to connect any two of them. For that, we have to do these four steps. First, we have to recognize the crime scene. We have to document the samples from the scene, collect them properly, and preserve it in a so that it cannot be contaminated. This is the story which was first used palynology in solving forensic problem in 1959. It is the example where first forensic palynology was applied. What is the story? The story is that in 1959, during a vacation cruise down the Danube River in Austria, a passenger disappeared from the cruise ship, and his body was not. They did not found the body of the passenger. They searched for his body. They did not find it. Then they searched his body under the water. Again, the body was missing. So police team came. They asked every co-passengers that what happened, how his body was missing. Then some of the co-passengers said that one person was talking that person who is missing. Maybe he killed him. So police went to his room, his cabin, and got muddy shoes. And the muddy shoes were not very usual. It was very unusual that in a cruise ship traveling, we never go to vacation wearing a muddy shoe. So police sent these muddy shoes to very famous palynologist Dr. Wilhelm Klaus at the University of Vienna. And the mud contained pollen grains of different types of plants which were growing around along the banks of the Danube River. But only one pollen grain was different, which is called alder, carrier, which is 20 million year old fossilized pollen grain, and its common name is hickory pollen grain. Then Claus searched for it, and he knew that that pollen is not possible to be in his shoes. It is in one location which is 20 kilometers away from the Vienna Danube River. So the suspected person was confronted with this evidence. He was absolutely shocked and he confessed his murder. And when the team went to the crime location, they found the body. Can you imagine that a single pollen evidence proved the entire murder story? That is the power of pollen grains. Also, in New York City, one magazine used to publish, which is called the Pollen Detectives, where all the cases that used to solve by the pollen grains, they used to publish in the magazine called Pollen Detectives. Like a major cocaine bust in New York City, murder case in Sweden 1959, Texas murder in 1959, 1995, murder in Illinois, USA during 1980, etc., etc. All these cases were not solved using DNA fingerprinting or serology or shoe print, tire print, or hand fingerprint. They were solved using the pollen grains. So, also, I am going to show you this video that in 2011, one Sherlock Holmes series came where Benedict Cumberbatch he was casted, and here for the first time in the commercial TV channel, it was shown that pollen grains can be used to solve criminal cases. How? Just you see the video. They have got a unknown pollen grain. They don't know which plant is it. So they have a database, software database, where if they put the image of the pollen grain, unknown pollen grain, the database will automatically match the pollen grain with the exact plant taxa. Here you see. So till date, it has not been done. It is just imaginary, but scientists are trying to do a database software which is called digital palynology or automated palynology, where the software itself will be able to identify any unknown pollen up to its taxa. This is the scenario in India. when I started my PhD in 2007, it was the scenario that anatomy, dendrochronology, palynology, ecology, limnology, plant taxonomy, these plant branches are not used in forensics in India. Only molecular biology, especially animal DNA. It is only used in forensics. And if you see this slide, you will be more surprised that till 2007 or 8, when I started my PhD, there was not a single publication on forensic palynology, but there were multiple, hundreds of publications from abroad, especially from New Zealand, Australia, USA, and UK. So I took this opportunity to initiate this branch of science in India. And I introduced some new tools or proxies in Indian forensics. Like, you know that we every day travel using different types of vehicles. It may be private car, buses, taxis, trains. I'm not going to mention train, but taxis, buses, private cars, auto, whatever it is, we take vehicles to reach our office, to reach our destination, to reach our home. And all the parts of the vehicle, they trap some soil, and that soil possesses pollen grains. But those pollen grain can be contaminated pollen grain. Only there is a one place which is the air filter of the engine of the vehicle. It only sucks the air, and all the pollen grains that are present in the air, that will be sucked in the air filter. So if you study the air filter of the vehicle, you can tell that what was the traveling route of the vehicle of that day or in last few days. So I did that experiment. Here you can see the red arrow. I took the air filter from vehicle. I took the soil from it, studied in the laboratory, identified the pollen grains up to its taxa, and I did some experiment. Here you see, so here you can see air filter can trap moisture, pollen, sand, dust, tire abrasion, and soot. Where it is going? It is going inside the engine. So here you can see all the pollen grains and dust is trapped in the air filter so that it can save the engine of the vehicle. That's why I took the air filter as my study object. And I published this article in Journal of Forensic Research from California, USA in 2013, where I introduced the air filter of vehicle to be a very important tool to detect the traveling route of the vehicle. Next, it is called the study of spider webs. You know that spider webs are everywhere, especially in the forests, in the old house, in uh, the places where people don't go. Their spider webs are present numerously. So if any crime happens, and if we get any spider web from the suspect's hair or cloth, we can study the spider web. And spider web is one of the most important vegetational indicator. Why? They are very sticky, they are very fine, cannot be visible in naked eye. We can see spider web, I know, but why we can see? Because of the dust. Otherwise, they are invisible. They are lighter than cotton fine. finer than human hair. So all the dust and pollen in the air, they get trapped in the spider web. And if we study the spider web, we are able to tell what is the vegetation type of the area, local area where from the spider webs were taken. If we take the spider web from the crime scene or the suspect, we can match the pollen proxy, pollen percentage, pollen types between the suspect, between the victim, and the crime scene. Here you can see this photograph. Every day we use some hair gel, hair spray, hair oil, shampoo. Other than that, when we shampoo our hair and we try to comb our hair, what happens? Our hair shows some magnetic property. This magnetic property is called the electrostatic force of the hair. And there are two types of electricity in our hair, which is called pyroelectricity and piezoelectricity. And that's why our hair can trap everything that comes to his way, comes to its way. So when you travel entire day and you are sitting near the window in a bus or in a taxi, your hair will trap more dust than your face because your hair has electrostatic force. What I did, I wanted to prove that hair can be used in the forensic studies. So I did some experiment. What I did, I sent some person from Siliguri to Rabangla, Sikkim, and vice versa. Some person came from Rabangla to Siliguri. One person went from Kolkata to Sundarban, and another person came from Sundarban to Kolkata. They traveled the entire day, and in the evening, they went back to their house. What I did, I told them to shampoo their hair. I took all the water and dust and everything in a tray. I centrifuged them. I make microscopic slides. I got some pollen grains, and then I identified the pollen. You will be surprised to know, you believe it or not, the person who went from Siliguri to Rabangla, I found pollen grains of so many high altitude pollen grains of the plants which grow in Rabangla. As well as the person who went from Kolkata to Sundarban, I found the pollen grain of mangrove plants in his hair who came back to Kolkata. So here you can definitely say that human hairs are very good proxy as a forensic tool. And also I published this in a journal in 2015. This is the real scanning electron microscopic photograph that I took during my PhD. Here you can see these are the human hairs, strands, and the pollen grains are trapped with the human hairs. I am just skipping because I have to shorten my time. Here are some limitations also of forensic palynology in India, especially. And if I tell you only the pros, if I don't tell you the cons, it will not be justified. There are some limitations to study forensic palynology in India. First, pollen study is not a very simple task. It needs expertise. A person should identify the pollen by its character up to its genus or tax species. If he identifies the pollen wrong, then he will not be able to solve the crime or the case. He can make a innocent to criminal or a criminal to innocent. So you should have precise and very top level expertise on forensic palynology. Second, for doing forensic palynology, you need a smart laboratory having all the modern facilities and modern equipments, aseptic conditions, and film chambers, all the major particles, which is not present in India till now. Third, you should have a pollen database of every state, every district, every region in your country. It is not possible for me. Someone calls me from Manipur or some calls me from Arunachal Pradesh or from Kerala that a crime has happened. You come and collect some sample and solve the problem by forensic palynology. Is it possible for me to do a database first, then identify the pollen with the modern database? It is not possible. So first we need to have a pollen database in our country of every region. We already have database in different countries. You can see European pollen database, PALDAT, the pollen database in other countries. Here you can see like Australian pollen spore atlas, the global pollen project, Missouri pollen project, like so on. But in India, there is no such database till date. This is a big missing area I should mention here. Last but not the least limitation is Indian judiciary system. Our Indian constitution, Indian judiciary have accepted the blood as a forensic tool, saliva, semen, vaginal swab, hair, bone, human skin, tissue, and DNA. But they do not acknowledge the pollen study to be a evidence in Indian court system. So that was the biggest challenge for me that after wasting my 5 or 7 years of PhD life, would it be wasted? Would I be able to do some contribute something to this field? So it was my aim. And if you know that in Kolkata Park Circus, there is a Central Forensic Science Laboratory, CFSL, which is under the Ministry of Home Affairs. From 2010 to 2014, 4 years, I used to visit the Central Forensic Laboratory. I tried to visit. They never allowed me to enter the forensic lab. I just told them that I want to meet the forensic biologist, Dr. Soma Roy. One day I got appointment and I visited her lab. I told ma'am that ma'am, I am doing forensic palynology first time in India, but no one is giving me any support. I want to work on real forensic cases. She said me that Sandeep, Indian judiciary system does not acknowledge pollen grain. You cannot get any real forensic case. I am not allowed to give you the case. So I was disheartened. But in 2016, suddenly one day, one fine morning, Dr. Roy, by herself, she called me and asked me, Sandeep, can you come to my lab today? I went to her lab. She told me that a 16-year teenager girl in Manipur, Imphal, she was gang raped and murdered brutally. And that news was aired in all over India that time. It was a very important and very popular case at that time that a very young 16-year girl was gang raped. When the case happened, it was in 2012. But ma'am called me in 2016 after 4 years. She told me that Sandeep, I am not allowed to give you the sample officially, but unofficially I am going to give you the sample. I want to know that we are not able to solve the case. What can you do? Just let's see. What she did, she told me that in Manipur, Imphal, there is a village called Luker village. It is a very small village. And in this village, the girl used to live. She was going to her aunt's house through a paddy field when she was stalked, she was stalked, and then she was gang raped and killed as well. In the crime scene, when police reached, police found blood, her nude body, her clothes, a rock which was used to kill the girl, and nothing else. Then the police investigated, and the neighbors told them that two boys used to stalk the girl from last 6 months. Police went to those boys' their house and collected two half pants and t-shirts which were muddy. So it was very unusual for the police team that why the muddy half pants and muddy t-shirts will be kept inside their almirah. So they collected those half pants and t-shirts. They collected the clothes of the victim girl. They collected the soil of the crime scene, the stone which was used to kill the girl, and sent all the things to the Kolkata Forensic Science Lab. Madam gave me all the samples. Here you see, A is the sellotape I used to scrape the soil from the rock. D2 and D1, these were the clothes of the victim girl. S4 was the clothes of the suspected person. N4, N6, N5, they were the also clothes of another suspected person. And the N4 was the cloth with some spider webs. If you observe carefully, you will be able to see that there is a spider web attached with the cloth of the suspected person. What I did, I studied all the clothes individually. I washed the clothes separately in glass beakers using the proper protocol, chemicals and acids. I collected the dust from the clothes. I studied. I made microscopic slides, observed under microscope, took photographs, measured them, identified them, and the result which came, it was really shocking for me. I am not supposed to tell the real data because I am not allowed to give the real names of the suspects, real name of the girl, and the real data percentage frequency or the statistics. Only I will show you these are the pollen grains which were present in exact number, exact frequency, and exact number of taxa in all the samples. Like the clothes of the suspect 1, clothes of the suspect 2, the stone which was used to kill the girl, and the clothes of the suspect 3, the clothes of the suspect 4, and the clothes of the suspect 5. Madam was really very happy that at least she got some result, which may not be effective, may be effective. She did not know. She just sent my result to the to the police team of Imphal. Police team, what they did, they confronted those two suspected people, and though those persons ultimately accepted their crime, that yes, we did the crime. After rape, we killed the girl. And finally, those boys were convicted for life imprisonment. So, what I did in my career, what I did in my research, I don't know. I have learned very little of my subject, but only satisfaction in my entire career is that by using my subject, by using my research, I gave justice to a very young teenage girl. Thank you so much. If any question is there.
Yeah, thank you so much, Shandip. That it is very fascinating one. Means I know you since 2009. I know what you were doing, and I know that.
Hello, am I audible?
Hello. Yes, I know that your work is going to give a great impact to the society, and we are very fortunate enough that we got you in this platform to share your work and the impact that you are
Hello,
doing for the society.
hello.
It's quite enormous. Thank you so much. Yeah, Anish, I can hear you. Please, you can continue.
Am I audible?
Yeah, you're audible.
Okay, okay. Actually, I have some technical difficulties. Sorry. Okay, so it was a great session, sir, and it was a very interesting one. আমার যেটা মনে হয় second sem and fourth sem, we all the students we will have that can connect themselves with the subject and I already told them about the topic a little bit, but they can clearly connect themselves to the topic, and they experience a great actually lecture. And I will request the students that if they have any question, though the lecture was extremely lucid, and it was very interesting. So still, okay, I'm requesting the students if they have any question, you can ask to Dr. More. And কেউ কি তোমরা পোল্যান্ড ডিরেক্টলি হতে চাও ফিউচারে, তাদের জন্য বলছি ইউ ক্যান আস্ক এনিথিং, এনি কোয়েশ্চেন, এনি কোয়ারি ইউ হ্যাভ, ইউ ক্যান আস্ক ডিরেক্ট টু স্যার, স্যার definitely answer. And also I am requesting all the participants if they have any question. uh,
Sir, am I audible?
Yes,
Sir,
yes, yes,
Hello, Dr. Deepan Bandyopadhyay, sir,
yes, please.
are you trying to
yes, sir.
speak?
Uh, sir, how can we make the
Yeah.
pollen calendar? How can
How
we
can we
make
make?
the pollen calendar?
Okay. Pollen calendar is calendar. We know that it gives us dates or months sequentially. So pollen calendar means, as we all know, that every plant does not flower every month or every season. Each and every flower has its own flowering month or flowering season. So if we take any area, or if I want to make a pollen calendar, every month I have to choose few particular days, maybe one day in every week, and I have to collect the flowers which are growing, the plants which are flowering at that time. If we collect the flower of present time, and if we go on entire the year, and if we make pollen photographs, observe them, measure them, if we can be able to describe them, like its shape, size, aperture, ornamentation, we can make a pollen calendar of the year. As well as, if it is also possible that we know that some pollens can cause allergic reactions in some particular area. Like in Kolkata, কিছু কিছু গাছ কলকাতার এই টাইমে বাতাসে ভাসমান থাকে, যেটা কলকাতার লোকদের অ্যালার্জি করে। আবার কিছু কিছু গাছ যেটা হয়তো নর্থ বেঙ্গলের লোকেদের অ্যালার্জি করে, সেটা আবার অন্য মান্থে হয়। তাহলে আমি যদি গোটা বছরেরই প্রত্যেকটা মাসের আমি অ্যালার্জিক পোলেনগুলোকে শুধু যদি কালেক্ট করি, তাদের পোলেন ডেটাবেস তৈরি করি, তাহলে আমি একটা অ্যালার্জিক পোলেনের ক্যালেন্ডার তৈরি করতে পারবো, এবং এটা পাবলিশ করে লোককে বলতে পারবো যে এই মাসে এই গাছগুলো আমাদের অ্যালার্জি করে, তার মানে আমাদেরকে এই গাছগুলো যদি গ্রো করে সেগুলোকে কেটে ফেলতে হবে, বা সেগুলোকে অ্যাভয়েড করতে হবে, বা ওই এরিয়া দিয়ে গেলে আমাদেরকে মাস্ক পরতে হবে যাতে আমাদের অ্যালার্জি না হয়। থ্যাংক ইউ।
Thank you, sir. Sir, I want to know that is there any task on the pollen calendar? Is there any database from any region?
Yes,
If you can,
we have already done pollen calendar on honey, pollen calendar of modern pollen, as well as I am already working on a project on dwarfs to South Dinajpur and North Dinajpur, Malda, Murshidabad. I am working on these areas where I am going to make the pollen calendar of allergic pollens as well as family wise.
okay, sir. Thank you.
Yeah.
Anyone else have any question to the sir? We have one comment in the chat box. Onish, Dr. Onish Bhattacharya, can you please share the comment?
Yeah. Um, the question is pollen do not decay in course of time as seen in honey.
Now, is
So
it question or statement? I don't understand.
it's a statement. I'm confused. It is a statement or a question. Dr. Ahmed Abdul Halim Khan, can you kindly clarify, sir? Hello.
Yes, sir. You
Hello.
can turn on your mic and you can speak. So I want to
Yes,
know.
yes.
Okay, okay. He want to know
So
actually. It's a
it is a
question.
question, I guess.
Pollen from honey, as I have already told you, that Pollen study, modern pollen is neopalynology, and fossil pollen is called geopalynology. So if I am studying pollen from honey, it is neopalynology, fresh pollen. And fresh pollen, it is just a plant cell, just like other parenchymatic cell or other any mesophyll cell or any cell. So pollen cell is a viable cell. It has water. So if the water due course of time, when it is going through the process of fossilization, it is first the water gets out of the pollen. It is getting dehydrated. If the pollen is getting dehydrated, then it is preserved. But when the pollen is fresh, it should if there is a appropriate environmental condition, that is pH, that is water level, precipitation, humidity, that will actually help us to say that the pollen will survive or not. But as I have already mentioned, that pollen wall is having sporopollenin, and that sporopollenin is very, very helpful to protect the pollen. Maybe the protoplasmic content, the nucleus, the ribosome, all the intercellular organelles may be destroyed, but the cell wall will definitely be survived.
Yeah, thank you so much. So, do you have any other question from the participant? Yeah, thank you. Any other
Thank
question?
you.
Yeah, Devjit, you can
Uh,
ask.
Any other questions from the participants?
No, if not, then there is nothing in the comment box also. I can see. If not, we can then conclude the session.
Okay,
Onish sir, Dr.
Devjit,
Okay, Devjit raise.
Devjit
Yeah.
raising his hand.
Do you want to ask anything? Okay, I
Okay, I
guess
guess
it
it
was
was
Sir,
accident.
accident.
হ্যাঁ
হ্যাঁ
বলো
বলো
sir,
বলো
বলো
what
তুমি
is
বলো
the process of making medicine for cough by only using Adiantum philippines?
My dear, I don't make medicine using pollen. I told you that pollen is can be used to make used can be used as a medicine or can be used to prepare medicine. That which is called latropalenology. It is another branch of palynology, but I am not doing latropalenology, my dear, and I don't make medicines also.
Okay, thank you, Devjit. So anyone else? I'm requesting the participants if they have any question. Okay, so can I conclude that?
Yeah, sure, Onish.
Okay, thank you so much, sir. Thank you so much, Sandeep Mure, sir, that you have shown us
Not
a
sir, Sondip Da.
okay, Sandeep da. না মানে you are having a hectic schedule for few days and your health is not also well but still you got the time and you have give us the opportunity to listen to your interesting and exciting actually research works and we are very fortunate and our students are also very fortunate to witness that. So thank you so much. In future, we will hope to have you as a offline mode also. So thank you so
Actually,
much.
I enjoy offline talks because, as you know, I give frequent lectures. I have already given more than 35 lectures, invited talks last few years. But I don't enjoy that much in online mode because face-to-face interaction is better than this. You know.
Exactly, exactly. I 100% agree with you. So thank you so much, sir, for being with us, and thank you everyone. Thank you all the participants. And now we will have a lunch break of 1 hour, and after that, at not 2 p.m., 1:50 p.m., we will gather here again, and we will have our paper presentation session.
Honest,
Thank you
I just
so
want
much.
to say something.
Yeah. Sure, sure, Dada.
Finally, I'd like to thank all of you for inviting me today. All the members of the organizing committee, head of the department, principal sir, and all of you who have organized this. Thank you so much.
Thank you so much. Thank you so much, Shantipanta.
Thank you so much. So we will meet at 1:50 sharply for the paper presentation. Thank you.
Okay, thank you.